to all the students. We are dealing with Unit 5, Coordination, the Linking System. Now animals we have completed. Now we are dealing with control mechanism in plants. So in the previous class we have seen how do plants respond to stimuli. Right. So there Mimosa pudica, that one it is touch me not plant, how it used to fold it. So we have seen. Right. Now here some of the plants they used to show their growth by taking the support, that is the tendrils of plants. And we have seen the plant growth hormones like abscisic acid, auxins, cytokinins, gibberellins, and ethylene. So these are the various plant growth hormones. Right. Now these plant growth hormones, they are very important, that it used to come for 4 marks as essay type answer. Right. Now let us come to the new topic, that one it is activity 5. Right. So in activity 5 we see the bending towards the sunlight. So here what you have to do, first figure you see it and the second figure also you see it. So here in this activity 5 what we have to do, so you have to take one glass jar filled with soil, right, and you sow a bean seeds. Okay, so near the wall of the jar, right. So then afterwards here you observe after 4 or 5 days you will be noticing that the seeds it used to germinate, right. So when it is going to germinating, keep the jar under the sun and you observe how the roots and shoots grow, right. So then afterwards here always you see the shoots show growth towards the sunlight. So already in the lower class you have completed that, you know it in lower class, that when in a box, okay, so in a box to one corner when the hole it is there, so you keep the potted plant in the box, then you observe, you keep it in sunlight. So wherever the light it is there, it bends towards the hole and it comes outside, right. So in the same way here also, that one is in the first figure, you see here it shows more growth towards the apical region, that one it is wherever the sunlight it is there, it shows a growth towards the sunlight. But when you again in the second figure when you see it here, that is then tilt the glass jar and keep the plant horizontally, right. So keep horizontally. In the second figure you see the pot it is horizontal direction. So when you keep it in a horizontal direction also, that here from the sides the root, that is the shoots it used to grow when it tilt also, when it is growing horizontally also, from the sides it grow, it shows a growth towards the sunlight. Okay now, so when you keep it for a week and you observe it, so all you got understood I think. So in your textbook they have asked question, that one it is, does the shoot take a horizontal tilt after a week? Yes, when you keep a horizontal, the plant it shows growth at the sides. Okay, so that is the side, it is mostly it is going to be exposed to directly, it is going to be exposed to the sunlight, right? So here that is whenever the plant it is going to be exposed to sunlight, so there the growth it shows, right? Now next one, let us see here. By this the phototropism, that one it is phototropism, it is the growth, the plants show growth towards the sunlight, right? So regarding this the Charles Darwin, okay, and his son Francis Darwin, they performed few experiments on the phototropism, right? So here this like this is so many experiments they used to did it. That one it is one, I'll tell it. Okay, so here this what they did, they cover the terminal portion of the tip of the stem with a cylinder of metal foil. So here this when the plant it is there, so the terminal part, the apex part, they have covered with the metal foil. Okay, so when they are covered with the metal foil, so then here they expose the plant to light coming from the other side. Okay, so they have here covered the terminal part with the metal foil. So the other side, wherever the sunlight it is there, so there it shows the growth. So like this they did some experiment regarding the photosynthesis. The very most important experiment that in 1926 the Dutch plant physiologist F.W. Wendt, okay, so he is a scientist, F.W. Wendt, he did a experiment, okay, so regarding the phototropism, right? So here this just regarding the phototropism, Charles Darwin and his son Francis, they did the experiments on the plants that it is going to be exposed to the sunlight by closing its tip with the metal foil. So other side it was open, so when it is going to be exposed to the sunlight, it shows a growth. So in the same way, botanist, that one is a plant physiologist F.W. Wendt, here he did an experiment regarding the phototropism process. I'll show it now. Now here the Wendt experiment, that one it is Wendt experiment, right? So in this Wendt experiment, let us see that one it is. What is this Wendt experiment? So in the Wendt experiment, what he did it, that he took oat plant, that one it is oat seeds, it used to be there, no? He sowed the oat seed in the soil, then after few days it produces the shoot and the root, okay now. So the root grow inside the soil and the shoot above. So that in the figure you can see that one it is the oat seedling, oats, O-A-T, oat, isn't it? Oats, it will be there now. So that oats seedling you are seeing, right? So here this oats, the below that one it is a root, and the above one, that one it is a shoot, right? So then afterwards what he did it, here he, when the shoot it is growing, he cut the tip of the shoot, okay, you observe it, the tip of the shoot, what he did it, he cut the tip of the shoot. Right. Then afterwards, he kept the tip of the shoot on the agar block, isn't it? So agar block, AGAR, agar block. So that one it is the nutritive medium, so which give nourishment. Okay. So where here this agar, you can see there agar block. So the tip it is there, no? So the tip what he cut from the shoot, that tip he placed it on the agar block. Okay now. So after 1 hour he placed it. Then he kept the agar block on the decapitated plant, that is stumps of the decapitated plant. Decapitated plant is what? That one it is the cut part, the shoot it is there, no? So on the head of the shoot he kept that agar block. Okay. So then afterwards, here when he kept the agar block, it shows the movement, the shoot away from the agar block, isn't it? So there you can see the bend. That one when he placed it, it shows movement away from the agar block. Okay. So then here why it is going to happen? That one it is due to hormonal signals, that is in the plants also the hormone it is the auxin mainly responsible for the elongation, cell elongation and differentiation of shoot and the roots, isn't it? So due to the hormones, that one it is due to the presence of these auxins, isn't it? So auxin, he kept this experiment in the dark. Okay. So he kept in a dark during the entire experiment. Right. So here this, then what he have observed, that one it is, it shows the coleoptile, that one is the stump, it shows the bending away from the agar block. So that one it is, here what he is telling, that it is the plants show growth towards the sunlight but in a dark also. Here due to the presence of the auxins that is going to released in the plants, mostly at the tip, at the apical region it used to be there. So there also it shows the growth. Okay now. So here that one it is due to the chemical stimulus, right? Some electric impulses it is going to be passed. Here you can see it. That is from the agar block, some electric impulses it is going to be passed. So that electric stimulus is known as auxins. Okay. So this auxin means to increase, isn't it? So to increase or the cell elongation of the shoot. Okay, got understood? Now here that one it is. Now let us see once the went experiment from your textbook, that one it is. There you can see that one it is the oat seedling. So down it is the root and the above the green color it is the shoot. So the first figure, that one it is the shoot with the root of the oat plant. Then the next figure, he cut the tip of the shoot of oat plant and that tip he placed it on the agar block. So small square like it is there, no? So that one it is the agar block. Okay, so when he placed the agar block, when he placed the tip on the agar block, so this agar block, from the tip of the stem, it passes the chemicals. That one, it is the chemicals through this agar block, and he placed that agar block on the third figure. You see it, he placed the agar block on the tip of the shoot, isn't it? He placed the agar block on the tip of the shoot. Okay, so decapitated shoot. Okay, so that one, it is a cut part. Then afterwards it shows the bend away from the agar block. So there also the growth, it used to be there. So why it happens? That one, it is the agar block. First you kept the tip region, cut part of the shoot, you kept on the agar block, so that from the tip of the shoot where the auxins it used to be passed, so from that agar block the auxins, it is going to be responsible through electric impulses for the bend. Okay, so this one, it was the vent experiment. It is very important, right? So ఇక్కడ వెంట ఎక్స్పెరిమెంట్ ఏం చెప్పారు? సో హియర్ దిస్ ఓట్ సీడ్లింగ్. సో హియర్ ఓట్ సీడ్లింగ్లో రూట్స్, దెన్ ఆఫ్టర్వర్డ్స్ షూట్. సో ఈ షూట్ టిప్ అనేది కట్ చేశారు, కట్ చేసి అగార్ బ్లాక్ మీద పెట్టారు. సో దాని తర్వాత హియర్ ఈ అగార్ బ్లాక్ మీద ఆ షూట్ టిప్ పెట్టినప్పుడు ఏమవుతుంది, ఆ టిప్లో ఉన్న ఆక్సిజన్స్ అనేది అగార్ బ్లాక్కి వెళ్ళిపోతుంది. దాని తర్వాత ఈ అగార్ బ్లాక్ తీసుకొచ్చి మళ్ళీ ఆ టిప్ ఆఫ్ ద షూట్ ఉంది కదా, అక్కడ పెట్టారనమాట. అలా పెట్టినప్పుడు ఈ అగార్ బ్లాక్లో మనకి ఆక్సిజన్స్ అనేది ఈ షూట్లోకి వెళ్ళిపోయి ఎలక్ట్రిక్ ఇంపల్సెస్ అనేది ప్రొడ్యూస్ అయిపోయి అక్కడ బెండ్ అనేది మనకి కనిపిస్తుంది అనమాట. సో ఇది అంతా డార్క్లో జరుగుతుంది. అంటే లైట్లో హియర్ ఫోటోట్రోపిజం, ఇట్ షోస్ గ్రోత్ టువర్డ్స్ ద సన్లైట్, బట్ ఇన్ ద డార్క్ ఆల్సో ద గ్రోత్ ఇట్ ఈస్ గోయింగ్ టు బి టేక్ ప్లేస్ బై రిలీజింగ్ ఆఫ్ ద ఫైటోహార్మోన్స్ ఆక్సిన్స్ అని నిరూపించారు. సో దిస్ వన్, ఇట్ ఈస్ ద వెంట్ ఎక్స్పెరిమెంట్. Now let us come to the next topic. So that one, it is trophic and nastic movements in plants. So what is mean by trophic movement? So trophic movement, it is the movement of the individual parts of the plant when it is going to be subjected to external stimuli. So this type of response is called trophic movement. So that one, it is external stimuli. That one, it is some plants, they may grow towards the sunlight. Some plants, they may show growth towards the water. Some plants here, that one, it is the roots show growth towards the deep into the soil. Okay, so like that, here some they require the support. So these are what trophic movements. Okay, so trophic movement. Now let us come to the next one. That one, it is the nastic movement. Okay, so the nastic movement, that one, it is the direction of the movement may not be determined by direction of a stimuli. Okay, so this type of response is called nastic movement. That one, it is the direction of the stimuli determines the direction of the movement. Sometimes the direction of the movement may not be determined by the direction of the stimuli. So stimuli and response, So sometime it cannot be determined. So that one it is called it as what? Nastic Moment. Now let us come to the phototropism. So what is mean by phototropism? That one it is the plant show response towards the light. Okay. So that one it is called it as what? Phototropism. Photo means what? Light. Tropism means movement. Okay, ma. Next one, geotropism. Okay, so Geotropism. Okay, so geotropism, there you can add geotropism also. Here, geotropism, that one it is. Here, the you might be seeing the plant, the roots grow downwards, isn't it? So that means the plant is responsible for positive for uh, gravitational force, isn't it? So this one it is the geotropism. You take example of the plant roots, it show movement, that is the plants grow downwards only, right? Now that one it is the geotropism. Now next one, that one it is the hydrotropism. So hydrotropism, that one it is the roots are always in one direction away from the rock or the wall where the water is available in the soil. So here this the roots they require what? Water they require it. So wherever the water it is there in the ground, so there the roots it move towards the water. So that one it is called it as water hydrotropism. Next one it is the Thigmotropism. So Thigmotropism, so here you take some of the creepers like cucumber, bitter gourd, the stems they are weak and thin. Okay now. So here that one it is which requires support. So they are what? Which require support, like when you take the bitter gourd or cucumber, so they may not grow erect like a mango or guava trees, isn't it? So they require water support. So that we call it as what? Tendrils. There the figure you can see the tendrils. We call it as what? Tendrils. So that one it is the tendrils, they are thin and thread like structure, and here that they grow on the leaves or the stems or they climb plants, isn't it? So they grow towards the support and wind around them, isn't it? Take money plant also. So they require water support, and when they get the support, they wind towards it, isn't it? So this type of response when in come in touch, okay. So that one it is called it as what? Thigmotropism. Thigmo means what? Touch. Okay, got understood? Right. Now next one, the last one, that one it is chemotropism. So where you can see this chemotropism, that one it is when you take the flowers. Okay. So the flowers here, that one it is the carpels of the flowers is what? Sweet, isn't it? Sweet, right? So here the butterflies they will be fluttering on the flowers for their nectar. So they'll be sitting on the flower for nectar, for what it is sucking the juices from the carpels. Okay, so that one it is carpels, we call it as what? Gynecium, right? So they used to produce sugary substance, right? So at that time this chemical substance stimulates the pollen grains which fall on the stigma, right? So here that one it is the pollen grains respond to this stimulus as pollen tube grow to reach the ovule for fertilization. So this type of response to chemicals, it is called it as what? Chemotropism. You take the better example, that one it is here chemotropism. That one it is the flowers, there here during the process of reproduction process, here that the male and the female flowers, it used to be there, right? So here what it is going to happen, that is when the pollen grain dust on the female flower, that one it is a gynecium. It contains what? Ovary, style and stigma. Okay, so these pollen grains, when it comes in contact with the stigma, so there the pollen grain, it forms a pollen tube, so that it is going to releases the carpel, that one it is the stigma, it releases a sticky substance. So that one it is a chemical, the substance it is going to release. So whereby the pollen grain forms a tube like, okay na, forms a tube like, and where the reproduction process it is going to occur. So where it is going to fuses with the egg. Okay, so this all you can go through in 6th lesson, the detailed study in 6th lesson. So just you know it, the chemotropism, when the pollen grains come in contact with the female, that one it is a stigma, gynecium. So there it produces the chemical substance. Okay na? So that one it is called it as what? A chemotropism. So this tropic and nastic movements. So this all you get for 2 marks or the 1 mark. Okay ma? So children, here go through the textbook. Trophic Moment, Nastic Movement, Phototropism, Geotropism, Hydrotropism, Thigmotropism, Chemotropism. Go through this. Now we complete our 5th unit. Okay. So here the writing part you do it and read the textbook thoroughly, right? So here I end up.